Hey everybody, welcome back to another episode of Public Access America. We're joined by our friend Dan, and I wanted to introduce this because I think it's an important thing to understand. Back in 2018, I met somebody on Twitter that was working for an organization called the People of Reason and Progress. He was the social coordinator, he was the executive of article, he was, that sucks. Anyway. Hey everybody, welcome to another episode of Public Access America. My name is Jason. We're going to be joined by Dan and Jeffrey shortly. I wanted to give you some background on Dan though to let you know why this episode's important. I met Dan back in 2018 where he was working as the social and communications director over at the People of Reason and Progress. It was a group of 30,000 scientists that were banded together to battle the misinformation of the day. We were doing a podcast at the current time. I had a co-host, Lexi, who was the original Jeffrey, and she wanted to discuss everything. And Dan wanted us to discuss everything. So we decided to work. He was collecting podcasts that would work with the People of Reason and Progress, and we were a podcast that signed up to work with them. Uh, that group, that organization did not pan out. It was a great organization, but it didn't work out. And I've been Dan's friend since then, and we've watched each other grow, and we've been together. Dan was a contract tracer in Pennsylvania during COVID. He as a National Guardsman, he went to hotspots all around the country addressing the situation, and he also stood and guarded our Capitol after 1/6. He's done so much more. He's got his food inspections certification, and that's cool. He's gone on to even get more degrees, and now he's starting something new, and we're going to talk about that next. So thank you. You can find Public Access America anywhere you find Public Access America, but I also want you to go over to Twitter and follow Epicenter Genomics. That's where Dan is now, and that is Epicenter Genomics LLC, a great organization we're going to learn about right here, right now on Public Access America. Welcome back to Public Access America. My name is Jason. His name is Jeffrey, and we are with Dan, one of my favorite people in the world. I've known him since 2018, and we have worked and collaborated on raising the intelligence of America and the world together, and I love him for that. He's got a new venture going on, and we are here to discuss that because I'm excited about that. Everybody knows Dan's origin story if you listen to the podcast, but here he is doing something amazing. So go ahead and go to Twitter while you're listening and follow Epicenter Genomics, and that is Epicenter Genomics LLC on Twitter. And we retweet their tweets so that I think people should know that's how important I think it is to the world. And Dan, I just want to get this started. We have some questions that we all collaborated on, and I just wanted to start with the most basic. What is genomics?
Yeah, well, first of all, thanks so much for you're very kind to say those nice things about me, and I've loved supporting the show, and I really appreciate what you guys do. So thanks again for having me on, and for me talking about something I'm doing a little bit. So yeah, so my background personally is in infectious disease epidemiology for people who haven't shown listened to the show for too long, and I periodically come in and comment on the state of COVID and monkeypox and other things going on. So recently, a couple of friends of mine, really great colleagues, and I started a new venture called Epicenter Genomics, which is a consulting company that's dedicated to using technology in a field called genomics to kind of advance the strengths and the abilities of public health, especially specifically for the surveillance and response to infectious diseases. So, genomics refers to it's a branch of what we call molecular biology that's focused on the study of genomes. Now, what is a genome? It's the complete set of like genetic code within like a cell or organism, right? So, humans, we have 46 chromosomes, right? Our genome is the entirety of those 46 chromosomes. For bacteria, I mean, depending on the structure that they have a lot less genetic material because they're just like a single cell. A virus, it's nucleic acid. It could be a DNA virus or an RNA virus. What it carries to encode or tell itself what to do to reproduce inside a cell, that is its genome. And so genomics is the study of those. right? You look at the genetic codes of these organisms, right? It can be a bacteria, a virus, or a human, or a cow, or whatever, and you study those in detail to have a better understanding of how the biology of that organism works. And there's a lot of different applications for this. The Human Genome Project was a really huge initiative and still continues to be in its own way. Genomics is being used to understand diseases like cancer, other congenital defects, trying to find mutations or specific genes that are associated with different conditions or high risk for cancer. But genomics is also being used in infectious diseases to study and track the trends of infectious diseases as they spread, as well as how those pathogens work, right? So try to better understand, the therapies that we can use, different antibiotic discovery. There's all kinds of really interesting stuff that's happening with genomics across the field of biology, and it's rapidly expanding. So that's kind of the science at the core of this new venture that my friends and I started.
That's cool. I like that.
Fantastic. I mean, you really kind of, with that, you really started to touch on what genomic epidemiology is. So, maybe just a quick, clear, concise definition of what that is.
Yeah, sure. So for the application that we use, as Jeffrey said, is genomic epidemiology, right? So think of it as the two different terms: genomics, right, the study of genomes, and epidemiology, the study of the spread and risks of disease. Right. In the context of infectious diseases, genomic epidemiology is the study of the genomes of these microbes, these bacteria, viruses, fungi, parasites, whatever, to identify when they are causing outbreaks and to track the spread of those pathogens. And that's been gaining a lot of steam in different contexts. But one kind of classic example that's become much more high profile is the COVID pandemic, right?
Mm.
All these variants that we talked about. We started with like alpha versus beta, B117, Delta, Omicron. All these new variants have been classified using techniques in genomic epidemiology, right? So being able to
Ah.
study changes in trends in what the genetic code of the virus has, right, what it carries, the different mutations that kind of accumulate over time as the virus evolves. And so you have these different variants that are classified by the shared mutations that they have that are unique to those groups, right? And the naming schemes, all the variants that get weird, like there's Omicron is a simpler version of the full names based on one type of scientific lineage, but it refers to a group of viral pathogens within COVID SARS-CoV-2 viruses that all share a number of specific mutations, and then the subvariants of Omicron they all have the Omicron's nucleotide mutations, whatever, and then they have additional ones that are unique to them. So that's how we go from something like A versus B to like B117, B11529, and the BA variants like B11529.4 and .5, right? It gets really complicated, but the main thing to emphasize is that all the discussion about the details of new variants of COVID coming out, and the news, how the new strains of influenza evolve over time. All of that is based in genomics and genomic epidemiology. So that's like kind of the classic example that's emerged in the last couple of years of how applicable this field is in science.
It seems like that's something that's been actually lacking since we've all started learning about viruses and Omicron and COVID and all this, is that we want to bury our heads in the sand and we don't want to know where it is, but then we're also not catching it early. So I think this is a vital thing that I think people need to know this exists. It's kind of like the weatherman. You kind of want a really good epidemiologist on your side, you know?
Yeah, absolutely. And there's some world-class epidemiologists that have, developed and refined these techniques in collaboration with, computational biologists and bioinformaticians. These people who have incredible like computational skills to study the evolution of microbes over time, and they have been working together for many many years. But especially with COVID, that's really brought them into the fore of their collaborating, performing these surveillance when COVID starts, coming to a new area or started coming to a new area in 2020. There were samples that were sent to, say, the CDC as well as to other private labs that did these analyses. The genetic material, the genomes that were sequenced from those, were able to be put in a domain where these scientists could collaborate. And so very quickly we had a very good understanding of what this virus was, what mutations it had, what kind of risk factors were associated with those mutations, kind of as compared to previous studies of viruses of a similar origin. And so it's been really important for us to emphasize that when new variants of COVID, for example, arise, it's not just scientists inventing something else to say, oh, well, we need to keep the public scared or whatever. Like that's kind of a common trope or a common response that I hear of people who are skeptical about these variants. It's actually because there's a massive infrastructure happening in the United States and worldwide to conduct surveillance and continue to sequence SARS-CoV-2 genomes and analyze them in detail and compare them to other genomes that have been sequenced elsewhere and see what the trends are. It's an incredible multinational effort. But again, it's not the only initiative that's focused on genomic epidemiology, right? Genomics has been used to track the spread of different variants of the influenza virus, which helps us inform how we do or develop new vaccines every year. It's been used extensively for food safety, right? The FDA has a whole genome tracker infrastructure that they use. Like, I think there was an E. coli outbreak just a few weeks ago that kind of hit the news that were in multiple states. Like whenever you see how did they connect like 60 or 70 cases of this bug for across multiple states, well, they did genome sequencing and compared them, right? Genomic epidemiology. And then in my specific example, my specific focus in my research has been on healthcare associated infections, right? You go into a hospital for surgery, you pick up a nasty infection while you're there. Using genomics to track the spread of those pathogens within a hospital to try and find how they're spreading and by extension how to stop that spread with better techniques in infection control. So the applications for genomic epidemiology are really limitless in terms of infectious disease epi.
Damn.
He answered my question. I was going to ask for specific examples to where genomic epidemiology actually saved the planet, and you went ahead and did that. I appreciate
I'm
it.
sorry, I went ahead of the plan. My bad, but
No, I love
I
it.
just get so excited talking about this stuff. It's really fun.
That's why you're my friend. That's why you're our friend, because you get excited about this stuff.
So, so I'm going to deviate just a little bit because I feel like
Yeah.
we're touching on something that is, I think, really important, and it kind of talks about it's kind of like how we've developed our vaccine policy and how we're looking at addressing COVID. But it also, I think, the question is with the rise of the new vaccine technology. The question is what link between your field of study, what the company is going to be doing, and new vaccine technologies do you think exist?
Hmm.
I think the connections are really strong, and it's not that we're not really inventing something new in this case because it's already been done every day, right? Pathogen surveillance and understanding how viruses and bacteria evolve over time is really critical to understanding how we can stop them and how we can be prepared for future variants to arise, as well as for therapeutics and vaccines that can be developed, right? There's a whole for example, with drug-resistant bacteria, right? A personal favorite of mine because it's a really serious public health crisis. There's a whole field of genomic epi that's dedicated to understanding the evolution of antibiotic resistance and looking at the genes that encode resistance or that can be shared among different bacteria. Which is a whole other process, right? But
Mm-hmm.
understanding how those, spread so that we're aware of the relative different threats of different antimicrobial resistance mechanisms in which drugs are potentially not going to work as often on different bacteria. So that's a whole subgroup of that. For vaccine development, right, with we talked about, I think you're touching on, Moderna and Pfizer's mRNA-based vaccine technology. But yeah, any kind of new vaccine, in order to answer the question of how do we develop a vaccine, you have to be like, well, what's out there? What's out there? What is the pathogen doing? How is it mutating? How is it evolving? And how is it causing disease by extension of those mutations, right? With COVID, with every new spike protein or mutation that we tracked over the years that this genomic surveillance has been going on, we've been able to follow this up with detailed studies in the lab of, well, is this going to be able to bind more effectively to a particular receptor in the cell and by extension make infection more serious or more effective or efficient, right? So genomics touches on a lot of different technologies because it gives insight into what the virus is doing in the real world. And then once we have a better idea of what's going on there, we can then take it into the lab, study the virus or the bacteria in the lab, and get a better understanding of what those mutations actually mean. And so there's this interplay between what we call wet bench microbiology, right, which is pipetting and biosafety cabinets and cell cultures and everything to study the how viruses and bacteria and pathogens work. And then what we call kind of dry lab, which is the computational bioinformatics approach of genomics. And there's a huge interplay where they kind of cross inform one another. And it's important to support that. But also, once we have a better understanding of what a pathogen is doing based on the joint collaborations and discoveries being made there, trying to push that into action, right? Now we know that there's a particular variant that's moving on, and now it's particularly nastier. We now know that there's a new drug resistance mechanism that's kind of emerged and is now spreading, right? What do we do in response to that? How do we communicate those findings in an effective way to public health officials, but also policymakers that don't have a particularly robust molecular biology or microbiology background, but they are responsible for, say, anything from public health to economic policy to the leadership of local or state or federal governments, right? So it's not just about what we can do with the science and how we apply that, apply it within our own field, but how do we take those findings based on genomic surveillance that we're doing and translate that into effective policy, into effective interventions, get people who are not staring at genomes on their screens of their computers for hours and hours a day. How do we get those people bought in and to collaborate with them in an effective way? That's the really key question of the future of genomics. And that's kind of the focus of the company that we started. We want to push this into action, intervention, policy making more effectively than just having this kind of surveillance going on or not potentially going on depending on the pathogen.
Fantastic.
I find that all fascinating.
That touched on so many things that I wanted to ask about, and it's like, okay, I was gonna ask. Nope, no, I'm not gonna ask that. No, no, I'm gonna ask that. So the real question, and this is something we've talked about before, is the struggle of public health messaging, and what sort of issues do you see coming up in terms of messaging with
epidemiology,
genomic epidemiology. I forgot the word that I needed to put in front of it, and so they're like sitting going
welcome to old age, buddy.
It's like walking into the kitchen and going, why was I in here?
Don't worry, I still do that, and I'm a fair bit younger than both of you. I won't say my age though. But it happens to all of us, right? But to your question about what are the challenges, right? I think the way that we conceptualize it, my colleagues and I, we think about it in terms of different languages, right? So there's a language of public health, right? There's the jargon, there's transmission rates and reproductive numbers and incidence versus prevalence rates and intervention strategies and different ways to control and stratify your research studies. There's the language of public health, right? And there's a lot of jargon. It takes a lot of time to really become proficient in that, especially with regard to infectious diseases, because pathogens spread, new things emerge all the time. You have to be able to relate to how that works in all kinds of different environments. We can think about, there's a sub language for hospital infection control, and there's a different language for food safety and food surveillance versus wastewater surveillance, right? There's all kinds of jargon that happens within those fields that takes a lot of time to become very proficient in. And that's important because we want people to be very proficient in those fields, right? Because we want people to have good and safe and disease-free lives. But when it comes to genomics, right, you think about genomic and epidemiology as the two operative parts of this field. Genomics has its own jargon and its own language that is in many ways fundamentally different, right? In genomics, we're thinking a lot about computer programming. We're using open source software, shell scripting, Bash, using R and Python, and programming these different alignment structures and phylogenetic trees and all this really complicated computational work that happens in order to analyze the multitudes of data, the gigabytes and terabytes of data that come from sequencing technology, right? And in order to become proficient in genomics, I mean, it's very difficult to be able to become proficient in genomics and become proficient in epidemiology together. It takes a lot of time to be able to speak both of those languages. It's like speaking two fundamentally different languages that are kind of in the same family, but not really connecting all the way. Like you have to learn different jargon, you have to learn kind of different grammar and where people are coming from. And the way that you view infectious diseases, right? If you're looking as a genomic or computational biologist, you're looking at potentially thousands or hundreds of infections at a time from all over the world or from a massive data set. If you're working in public health, you might be looking at a scale of maybe as most a couple of dozen infections in a local outbreak, right? So there's different languages and different perspectives that people in public health who work in infectious diseases come from on the different sides of genomics and epidemiology. And so the real challenge is connecting those two, right? We need, people who are proficient in both kind of sets of languages who can basically be interpreters between the two, who understand, who've worked in public health, who understand what it means to do case investigation, outbreak response, and program management, and really understand transmission dynamics of different infectious diseases,
Yeah.
and also understand why you are bootstrapping your phylogenetic analysis by 100 versus 500 versus 1,000, or why you're using this particular programming software for your nucleotide sequence alignment as opposed to a different one, right? And so when you put those people down in the same meeting room, a lot of times they talk past each other because their priorities are different, their jargon and the language that they're brought up in is different, and so sometimes things just don't get done, not because of lack of interest, but just of lack of connection. And so the real challenge in the field is being able to connect those two fields together in productive and meaningful ways that lead to interventions. And the linchpin, quite literally, in my opinion, is the people who can speak both need to speak both. They need to be able to understand genomics and epidemiology, be there in the meeting room, make stuff happen, to be able to help the genomics people understand the goals and desires of the public health people, and vice versa. So again, that's a major focus of the company, is not just being just genomics people who always just look at pathogen genomes and study the evolution over time, or just being siloed into public health departments or hospital infection control, but actually bouncing between those groups and relating to both of them and being able to connect the different jargon that those groups use so that everyone understands what everyone else is talking about. And then that's when magic happens. That's when interventions happen. That's when people stop getting sick because of new technologies emerging, when people are able to communicate and connect with each other. And we need the people who are the true genomic epidemiologists who speak both languages to be those people in the middle making that happen.
So you're going to be a funnel point, a communication funnel point in a way.
Yes, that's the goal, right? There's private companies that are developing all kinds of amazing technologies and diagnostics and molecular assays and things like that to identify infectious diseases. There's hospitals and health departments that need the answers what those companies produce. And then there's academic medical centers that have incredible excellence in this genomic surveillance, right? And a lot of times those groups get siloed
Hmm.
because of their various different priorities.
Sure.
And when you don't necessarily learn about what a single nucleotide polymorphism or a SNP is when you are doing hospital infection control, because it's not really relevant to your training. You're focusing more on how to perform your surveillance based on you're told that this is the particular pathogen that happens and being able to respond to that, understanding risk factors, being able to communicate with hospital staff on how to perform proper hand hygiene and proper cleaning, disinfection, and sterilization techniques. There's a whole skill set there, but that does not really include genomics because it hasn't really needed to in order
Hmm.
for it to be good enough now. So now, if you want to incorporate this new technology between like companies and hospitals and health departments and academia, right? You need to start connecting those folks and being the funnel of information, but also being the facilitators. I like to think of myself as like those old fashioned phones that you call the operator, and the operator would physically connect one line of a wire to one phone line and connect the other one, right? Making those connections happen and kind of supporting the conversations, and as well as being the subject matter experts in both of those fields. So it's a multidisciplinary approach. We have multiple goals that kind of overlap in terms of like educating and translating and like optimizing these partnerships.
A lot of
But I think it's really
translating.
important.
Yeah.
Yeah, it's really important because there's a lot of applications for this, anywhere from the single hospital level to like international surveillance of pathogens, right? Being able to, there's the same kind of goals at all these levels of scale, from the very small to the multinational.
Shit. That's a lot of data, Jeffrey. That sounds like your bag.
Yes, it is. And that's the thing is that when you look at it from a data perspective, it's like we can't exactly talk about answers if we can't really show it. And even like, because essentially what Dan is working on is essentially what I kind of went to school for, but more in a business sense than an epidemiology sense, is that you've got one person working on policy, who is, they're trying to make whatever rules and regulations are going to help make sense. You've got
Hmm.
the company who is, trying to figure out how do they meet their needs. And then you've got the general public who, is trying to get X, Y, and Z. And oftentimes, marrying those three is very difficult because, your corporate person doesn't necessarily have a whole lot of time to hear what it is you're trying to say, because they're like, bottom line, how does this affect the company? The
Right.
public, oftentimes they don't have the ability to understand, what it is that you do. Like, I explained multiple times what it is that I do, and people genuinely like start to glass over. And then you got the policy where they're like, that's great, but how does that help me? What do I need to do? And that
Right.
communication gets real difficult real fast. And so, like, even like in my field of risk, you know we are the ones that start to see the emerging trends before policy ever catches up, before corporate ever catches up, before the public even knows what's going on. And trying to marry all of those interests into one, you know group, one concise, one message that can be easily understood and interpreted by all three parties
Hmm.
is very difficult. And so, like, I can understand exactly what Dale's going for, because like it's not easy to try and communicate three separate ways what's happening.
Yeah. Well,
Yeah,
he's always
especially
been,
because it.
you've always been
Uh,
the communicator. You've always wanted to find a way to communicate all this to all people. So it makes sense that you're doing this.
Well, thank you. I appreciate that. I think to Jeffrey's point as well, it's not just about the jargon and the where everyone's coming from and their backgrounds and everything. It's also about the priorities everyone has, right?
Right.
Academia is going to have different priorities than public health is going to have different priorities from hospital executives versus,
Hmm.
policymakers, right? Everyone wants to know what the bottom line is for them and what it means. And oftentimes, when you come from a different perspective and have different goals, sometimes those don't translate into the best outcomes for everyone, and that's not anyone's particular fault. But there are definitely opportunities for, especially for the field of genomics and infectious disease epidemiology, for those connections to be more robust, so that the innovation that is happening in this field is actually kind of comes to fruition and comes full circle, because there's been incredible advances in the technology of genomics in the past 15, 20 years, right? We're orders of magnitude, many orders of magnitude more efficient and more cost effective at analyzing microbial genomes than we were just 20 years ago.
Right.
I think in early 2000s, getting a full bacterial genome sequenced based on the technology that was available then would be tens of thousands of dollars. Now you can do it for like 70 in a couple of days. And with that massive advancement in the strength of that technology, what is holding people back from leveraging that most effectively? And the answer is, communication and
Yep.
engagement, in our opinion, right? How do we get people to engage with this work, understand its value, and be able to adjust and evolve their policies and their education and their training to at least be familiar with the advantages of this new technology? Doesn't mean you have to become fluent. You don't have to take every infection preventionist in the country, every epidemiologist who works in the public health department, and make them experts in genomic analyses of viral pathogens, right? You don't have to do that,
Right.
but at least get in to appreciate the value added of this technology, and have the policies be evolved to incorporate this more effectively, so that there's a win-win around, right? And genomic surveillance is much more sensitive, it's very effective, there's a lot of benefits to it. Even though it does take time to learn how to do it, you don't have to get everybody to know how to do it. You just have to get people to appreciate it, want to implement it, and have that buy-in to translate it into effective policies. And that is our goal as a company, right? Be those conduits for those communications to happen. And whatever the barriers are to that happening, is it, my company doesn't know enough about what genome sequencing is, or we haven't had these productive conversations with this particular hospital C-suite, right?
Mm.
Being able to fix those issues just to let the stream of information flow naturally, so that there's a really good policy outcome at the end, and genomics is fully integrated. And then at the end of the day, fewer people are getting sick and fewer people are dying, because surveillance is more effective, response is more effective, because of
Yeah.
this new technology really being leveraged.
I think that's the point that the public wants to hang its hat on and it misses, is that people, if you could communicate this stuff better between departments, between silos, these pandemics aren't pandemics, right? They're caught early and often. and that's what you're working to do. You were talking about genomic surveillance, and I think about testing sewer water, right? Testing wastewater to find polio, and I think that the infrastructure being there. So when you say surveillance, do you mean like hospital surveillance or public surveillance like that?
So it depends, right? Surveillance is a general term that's used in public health to just like we want to see what diseases are out there for what particular time,
Right.
right?
But
And surveillance.
what's your eyeballs for that?
So it right now a lot of times surveillance and before the era of genomic surveillance was predominantly based on people getting sick with a
Right.
particular pathogen, and then you do some testing to try to figure out what it was. You would have to do all these wet lab techniques to antibody assays and biochemical assays to see, okay, well, you have this particular bacteria. You look at it under a microscope with a gram stain. Well, it's gram negative. It coagulase positive, catalase negative. Right? There's all these like
Yeah.
biochemical molecular assays that you can identify, and then from there you'd be able to identify or confirm what species it was, or if it was something new, or it was something old with a new twist on it from a new mutation. Right? And so that takes time, that takes effort, and it's still used very predominantly today. But that can be used at any scale, right? This is similar technology was used, at an individual hospital. Someone gets sick, well, you send a sample down to the lab, they analyze it, right? The CDC and the FDA and all these, the three-letter health agencies in the federal government, they have labs based on the same technology to identify these pathogens where samples are sent to them, right?
But
And
it's
now it's getting interesting where you can start using genomics as either a supplement or as an alternative to that, where you can get an answer fairly quickly and also much more sensitively, right? You're not just talking about, is this E. coli versus Klebsiella? Is this SARS-CoV-2 versus influenza? It's like, okay, now within a couple of hours or a couple of days, depending on which technology you use, I have an idea not only just what pathogen this is, but look at the mutations it has and how similar this is to another pathogen that was identified in 3 states over 2 weeks ago. Wait a second, there might be a common source for this, right?
right.
There's the evolution from the biochemical assays to something that we called pulse field gel electrophoresis or PFGE, where you take a genome and like cut it up with enzymes, and then there'll be like a barcode that you could kind of run out on a gel, and like you compare barcodes, right? Much more advanced, but still less sensitive than actually looking at like nucleotide by nucleotide ACTG, having that level of sensitivity. So genomics is definitely not going to permanently, sideline these other diagnostics, in my opinion. I don't
Mm-mm.
think that's going to happen, but it is a really useful tool with which we can evolve our field of surveillance, which can be at a hospital level, it can be at a county level, it can be at a single, building sewer water level or a single cooling tower for Legionella, or it can
Gotcha.
be national and international, right? It's not that there's multiple definitions of surveillance. It's the same idea of like looking out for pathogens that can be causing disease. You just have to adjust your scale based on what focus you have in particular.
Sure. When you think about surveillance infrastructure in the public, you want that. But it's what you're saying sounds like people need to go to the hospital, get tested, they find something, and then it's addressed. It's in real time, it's not predictive. You know what I mean?
Yeah, it's not predictive. I don't think there's some aspects of genomics where you can kind of predict to see what might be going on, and there's a lot of research happening into using past data from pathogens to see where the next mutations might be, and that's actually was pretty relevant during COVID.
Right.
But I think what's really powerful about genomics, especially in the last few years, is that there's been technology that's come out where you can go from dipping a bucket of into a sewer to getting a full what's called metagenome, which is just like whatever the genetic material is in your sample.
Hmm.
You can get that done in a few hours to maybe a day, and having a pretty good accuracy and a good understanding of what's hanging out in your wastewater, right? So you can go out and do this active surveillance at a much larger scale than you could before because of this advance in technology, as well as the
Right.
recent advances in data storage, right? You think about where we were 25, 30, 40 years ago, how many dollars per gigabyte would it be to store data? Genomics is very data heavy, right? There's a like, for example, my master's thesis, I was looking at like 3,000 bacterial genomes from a single hospital and the total raw data of those genomes was like 15 gigabytes, which by comparison, if you download a movie for streaming on your laptop or whatever, not that we do that anymore with like Netflix or Amazon, right, that would be like 2 or 4 gigabytes of data, right? That just gives you a
Mhm.
scale of how much there is. But because of the advances in technology in computer science and data storage, as well as the advances in actually, developing the technology to actually do genome sequencing, these are kind of converged where it's now much cheaper, much more effective. There's now a sequencing platform that's the size of a cell phone that you can fit in your pocket, costs $1,000, right? And you can use its technology. I've done it before. You go from looking and pulling a sample to sequencing it in like a day, and you can have a really good idea of the genetic content of whatever pathogens are in your sample in hours to maybe a day, as opposed to weeks to months of assembling a single really purified genome, which is where we were in the late 90s, early 2000s. So incredible advances in technology that need to be implemented in order to really propel public health into the 21st century. And how do we do that again? By connecting the interested parties to get them to realize the common ground they have and adjust their goals and implement this technology so that this more accurate, more precise, faster surveillance method is more available for their goals to ultimately stop people from getting sick.
Agreed.
Absolutely. So what is the bottleneck do you see, like, with that your company is, hoping to address and
Great
hoping to
question. Yeah.
alleviate?
Yeah, I think a lot of it comes down to, like I mentioned before, the education piece or the translation piece, right? There's a lot of people out there who are, frankly, we're comfortable in genomics. We've published a bunch of papers and have a bunch of academic training in genomics, right? There's a lot of people who know more than we do about how to really analyze and develop new software and evolve software to have a better understanding of what bacterial or viral or fungal or parasite genomes are telling us,
Hmm.
right, when we analyze them. That is there's not really a need for us to forge new ground of that because there's experts who are doing so much more than we ever have already pushing that really far forward. Similarly, there's a lot of experts in public health surveillance and public health departments and in hospital infection control that are making incredible strides in their policy making and their education piece, right? But like I said, those groups are kind of siloed. There's not a whole lot of overlap. It's starting to happen, but now we want to really break into the markets where genomics is not really as mainstream, right? How do we stop genomic or not stop genomics? How do we get genomics out of the major like tertiary hospital academic medical centers and the state health departments and the federal health departments, right? Or the federal, the NIH, FDA, whatever, CDC. How do we take that from being mainstream there to going to, individual small community hospitals and making sure that they are able to access these new advances in technology, right? What bottlenecks exist there? A lot of times it's education and awareness, not just training in how to do the genome sequencing technology. You don't need everyone to be trained up in that, but you do need people to be aware and appreciative of it, and to be aware that you can incorporate this into your work. And how do you connect? Well, you have a lab doesn't really have any sequencing technology. Do you want it involved in your lab, or do you want to start having a partnership with another organization that can do sequencing for you? How to connect those resources together, right? So that problem solving,
Wow.
de-siloing these other groups that are essential to public health that happen to be like at the local scale, individual hospitals, individual universities, companies that do wastewater surveillance, local public health departments, that if they wanted to do genomics, they would have to engage these higher up groups, and it would take time. And the reason why I know
Right.
this takes time is because I've been there, right? I have worked in a local health department during the COVID pandemic and had to struggle and beg, borrow, and plead just to get a single genome sequence up the chain to our higher headquarters to try to get it back. And by the time it came back, right, it wasn't really useful anymore because the outbreak had already kind of come and gone. Right.
Yeah.
So I've been there. And my colleagues have also been there, that we've worked in hospital infection control. We've worked in like I have a background in the military public health. We've worked in local government public health organization. One of our co-founders is a clinician. He's trained as a physician to care for patients, right? We've seen where all of these challenges could be addressed with better genome sequencing technology, whether or not it's actually the technology in that particular location, whether or not it's better connections or communications or policies connecting groups on the ground caring for patients and the health agencies that can do some of the technology. Right? We've been there, and we've struggled through these frustrating elements that we've kept hitting brick walls on. And so, having been on both sides of having incredible access to genome sequencing technology to train ourselves up on microbial genomics, as well as being on the side of public health where we know where those could be better implemented and seeing which opportunities are being missed, now we have a really solid idea of how to de-silo those groups outside of the federal level, like thinking locally, thinking individual hospitals, individual health departments, right? Those that we have that experience from both sides to be able to chart a clear path forward for different agencies because of our collective experience.
Wow. I think to myself, all of these people during COVID that thought they knew what a vaccine was and all the rhetoric going around. And I used to tell people there's thousands of professionals researching this. You don't need to have the opinion. You need to support the voices that have the information. And this just reminds me that no matter how much I try and learn about vaccines, you're always going to know more. And I appreciate you doing that so I don't have to.
Well, it's not just about who knows more. It's not just about the knowledge piece. It's the buy-in and the engagement from different groups because
Right.
this implementing genomics in these places where genome sequencing technology has not really been used is a steep learning curve for a bunch of different reasons, right? And what we really pride ourselves on is because we have this multidisciplinary experience, we haven't just been siloed into pathogen surveillance with genomics, staring at computers for hours. And then, although I did a lot of that for my thesis, and Mustafa, my co-founder, definitely did as well. We have that experience. We know what it's like, but we've been in the meeting rooms with multiple entities of different backgrounds, and we've been able to make those connections before, and really bring people together from the hospital side, the pathogen surveillance side, the local health department side, as well as the computational component of it to make these happen. And our CEO, Alex Sunderman, has been an amazing asset to make those things happen. He's made some incredible discoveries. So even though this is a new venture for the three of us to kind of embark on together as a leadership team, it's not new to us, right? We have worked in these multidisciplinary environments. We have worked in different settings. We have been able to kind of compare our genomics training to the implementation of public health, and now we can bring that all together with a combined mission and focus and vision of this individual company. So the company's new, but our experience certainly is not.
Agreed. That's why you've been on the show for 4 years. So that people would know you're the voice people trust. And I wanted to ask, is any of this information that you're creating going to be public facing?
Yes. So we're engaged on social media. We do a lot on LinkedIn and Twitter. So we want to keep people posted on what's going on there. But there's obviously when you have a private company in business or whatever, there's going to be times where we have non-disclosure agreements and you're working with companies with proprietary data and information, right? So we can't just like publicize, hey, we're working with everyone, X company doing Y thing for Z reason, right? We can't always be like that. But
Right.
it's definitely important to us to have this public conversation because our goal is to be the springboard for genomics to get out of kind of the niche multinational federal big budget surveillance gurus and into the mainstream of public health for the rest of the country, right?
Yeah.
And so a lot of that we still publish papers. I actually just had a preprint come out that's based on a master's thesis that we're going to post and share, and we still continue to publish our work on the academic translational research side, and we want to promote other voices in the field. So the work that we do, we might not always be able to be super public about, but we can definitely continue to be those voices and advocates in the public domain for people to appreciate this technology, for people to understand what our mission is, why it matters, and how it can innovate public health, and what it means ultimately for the consumers of public health, which is the public that is protected. Right.
Right.
So yeah, I fully agree and I fully endorse wanting to continue to elevate the conversation about this. And that's why I'm grateful to be on the show today, because you guys are giving us a platform to kind of share this information in a really valuable way. And I'm very grateful for that. But yes, we want to continue that trend of public engagement and communication and elevating voices, as well as obviously being a successful business.
So you kind of talk like about who you're, the fact that you're scattershotting your target audience. I mean, that's really the best way to talk about it. But I think in the end, still, like, one of the biggest problems that we faced during the COVID pandemic was the general public. Not
Correct.
and that, we had messaging that was constantly changing. We had messaging that was constantly contradictory. We've, and I mean, also to just the general lack of understanding of science in general
Mm-hmm.
with the general public. Well, they said one thing, but now they've changed their mind, and so, it's all just politics. And of course, for me, that's my fear as a scientist as well, is that the general public doesn't understand that when we have our original set of data, oftentimes we're trying to get our information out as quickly as we can so we can adapt whatever it is that we're doing, and it's only through getting more and better information, we change our messaging, which of course, unfortunately, when you have the messaging that comes from public health, it often is just like, oh no, we've done this and now we're going to do that, and not really speak to why it is that we're changing the messaging. So I guess in all of this too, how do you plan to bridge that gap to the general public?
So I think it's an indirect path. I think it's a very important question to address. I do want to say, for the purpose of this company, right? Our goal is, we are essentially a consulting firm, right? We are trying to work with other companies and stakeholders to try to facilitate the execution of genomics and in infectious disease public health. So that is the
Right.
goal specifically of the company. That said, I think the outcomes of that are going to have, if done successfully, will have really good ripple effects to the general public, because, something I mentioned before, genomic sequencing technology is an extremely sensitive method of surveillance relative to others, right? If you think about, talked about, you can look at nucleotide by nucleotide. What is the bacterium looking like, right? What is the virus looking like? As opposed to, oh, it looks like a flu virus, it looks like a coronavirus, it looks like a E. coli, Klebsiella bacterial strain, or whatever. By having that technology be more or better implemented, and having better resolution of those pathogens as they emerge and as they're kind of caught by genomic surveillance, that is going to in turn help public health infrastructure have a much more accurate understanding of what's going on very early on. And I think the classic example that's most recently relevant is the monkeypox outbreak, right? I've kind of raised, rung my own alarm bells about monkeypox, and it looks like cases are continuously or consistently declining for a few weeks. But I think what's really important to emphasize is that, because of the emergence of this new pathogen, some things that went really well was the genomics piece of it. We had very quick turnaround times for people at the international level identifying these new strains of monkeypox, sequencing them, analyzing them, and having a good idea of where they're at relative to previous monkeypox strains. And having that better information, we now know it's like West African clade monkeypox as opposed to another type of monkeypox, right? Having that better information early on from genome sequencing technology and how it's implemented helps improve the messaging by giving people a much more precise picture of what the pathogen is that they're trying to look at. And so if you're able to implement that at a local scale, individual hospitals, in individual health departments more effectively, then that's going to build trust, not necessarily at the CDC informing the president and forming their, the head of the NIH who's giving national briefings, right? That genomic infrastructure is already kind of there. But if you think about the local health departments, those are the workhorses of public health in this country. The individual hospitals, the community health centers, the wastewater treatment plants, right?
Right.
If you're able to arm them with more accurate surveillance technology through genomics, then they have a better idea of what their flaws are in their preparedness, prevention, and control and response systems, or how they can more quickly detect what's going on in the community so they can act act faster, or they can provide a clearer, more concise message with more detailed
Yep.
information to their community. And this is something I've talked about kind of on the more political side, right, which I don't want to get into too much now, but I firmly believe that if you think about local politics, that's where 90% of your life is affected, right? Your local regional government. I believe the same thing about public health, right? Local and regional public health, your local hospitals, your community health centers, your county health department, your wastewater treatment plants. Those are the entities that are really having a major influence on the protection of your health as you go on your day-to-day lives. And
Right.
so if we can bridge genomics more effectively into those spaces, into those domains, as opposed to keeping it with the big budget organizations that generally do much more widespread surveillance across the country, I think it's going to help the public trust health infrastructure more because of those local level communications that come as a result of it. So, we're not going to create ourselves as the messengers for this, but if we can start making better connections and making things more efficient at local levels for these different entities in these different communities, or get companies that have amazing technology to implement their technology in these different communities, it's going to help people at a local level much more effectively than if we just tried to be another like blog or resource or another analytics company that's just pumping out data. Right? We want to be
Right.
those force multipliers by making those connections more effective and more efficient so that the public are better informed what's going on in their communities.
Right. You're going to help the local infrastructure pass that information up in a way that communicates up the ladder what they're
Yeah.
looking for and what the
And,
urgency is.
yeah, and
Okay.
if solving those problems means going into a hospital and educating a bunch of infection preventionists on what is genomics, that's great.
Right.
We're happy to do that. If that involves talking to local health department leadership about different ways that probably maybe making a budget change to hire a bioinformatician to do some local level surveillance, that's great. If that means that a local health department needs some specific support on bioinformatics surveillance, like, hey, we have no one else to do this, can you just help us analyze what's going on for this problem, because we don't have an idea of what's going on, just we have some genome sequences, we don't know what to do with them, help us. We can
Right.
plug in and be that short-term response and say, give the man a fish, right, and then teach the man to fish afterwards, and helping them be self-sufficient afterwards. That's going to make a huge difference if we can go project by project at the local level. And so that doesn't necessarily mean we just are only going to work with local level clients. We want to work with large companies, and if state or federal departments want to work with us too, that's great. But I think that is a really great opportunity that I don't think is being fulfilled as effectively as it could be, and we want to be the difference makers in that realm.
Right. Again, because an infection starts small, and if you can catch it locally, you have a better chance of containing it. Right?
Absolutely. Yeah. If you
Okay.
have better surveillance early on, I mean, that's again, that happened with monkeypox, right? Monkeypox has gone global. There's no doubt about that. But we caught it pretty early on. There was
Right.
very rapid response. And now we have a good idea of how things are going internationally because the surveillance infrastructure was there. And it seems like the response and prevention infrastructure has built off of genomic surveillance really well, right?
Right.
We have really good public health messaging. We had a really good understanding of where monkeypox was hitting in terms of the demographics that were getting most disproportionately affected. Having a better understanding of why that was happening, part of that equation was you know, understanding, hey, there's a new strain of monkeypox that's kicking around and it's actually kind of similar. Where did it originate? Where has it gone? Where has it not gone yet? Okay, well, maybe this part of this country has not been affected by monkeypox yet because of the surveillance we have. Let's
Right.
educate and let's be preventative and proactive so that eventually when monkeypox does hit that location, they're a little bit better prepared. That has gone pretty well. Has it gone perfectly? No, nothing in public health does. But if genomic sequencing and genomic surveillance can be better, then processes that are downstream from that surveillance are going to act faster on better information so that they can be more effective in their interventions to prevent and control disease transmission.
Correct. A virus spreads because people aren't communicating with each other fast enough.
In part, but yes, absolutely.
And I think that's one of the biggest things is that it's that whole how do we go from, oh my god, this is doom and gloom, the end of the world pandemic, to hey, we have a localized infection that seems to be spreading. Here's the genomics.
Right.
Here's how we understand the spread. Here's how we need to be working at this from a public health level, from a hospital level. And
Mhm.
being able to have that communication of letting other people know, hey, we're seeing this rise, check your local communities around here, here's the standard messaging that we're working with, here's the standard data sets that we're working with. you
Right.
know it's one of those things. Like, I think all of us went, oh shit, it's the end of the world when we saw COVID. But when it came to monkeypox, everybody was so jaded from that they basically just didn't want to have that same level of response. And I won't say that it's been lackluster, but I will say that it's certainly nothing compared to what COVID looked like.
Yeah,
And
it didn't get the press that
but
COVID got.
but also too, part of it is that when you look at monkeypox versus COVID, it's number one, entirely different disease. Number two, very while there's some similarities in mechanism of transmissibility, it's not quite as doom and gloom. And I think it's just the biggest thing is like figuring out how do we not end up with that whole, like, either we go to the extreme of, oh my god, we're all dead, to whatever, fuck it, if it gets me, it
We
gets.
catch it early. People freak out when it gets too big, I think. And I think with monkeypox, it was addressed every day in a meaningful way, through messaging and through vaccinations. And I think that calmed the public down because we caught it early and people communicated better about what it was and how it was transmitted. And it was the whole messaging thing that COVID didn't have. COVID had an anti side to it, then the monkeypox didn't.
Yeah, I think there was definitely kind of a monkeypox skepticism, or hey, they're just creating something else to
Really?
keep
Mhm.
establishing the new world order. Yeah, there's I've seen a lot of messaging that's kind of been monkeypox is a scam, but I think and there were definitely some clear there were definitely some clear advantages that we had in the fight against monkeypox. Right, one was we did have vaccines that were at least moderately effective,
Hmm.
and we had a strategic national stockpile. The another thing was that there were lessons that were learned from COVID because from like the early response side of things to try to keep a kind of containment versus a mitigation strategy. Has that worked perfectly? No, but it's been, I feel like in many ways it's been a lot better. But I think another really important piece of it is that we were very quick on the draw for understanding that this new virus or this new strain of this virus had emerged out of West African, its home geographic location, right? And a lot of that had to do with, hey, there's samples of this new disease coming in. Let's get a genome sequence on it. Let's see how it compares to other genome sequences. Let's have availability of data
Yeah.
to be able to make these comparisons internationally, right? And it's been a lot of that infrastructure has been built a lot because of COVID, because in part genomic epidemiology has become much more mainstream in public health, at least at the, and like I said, the international, state, federal levels,
Hmm.
as opposed to the local and state levels, right? Most individual hospitals, most local health departments, whatever, even some of the states are not really as engaged in genomic technology for pathogen surveillance, monkeypox or otherwise.
Yeah.
Um,
Yeah.
but there was enough like international collaboration for us to detect what was going on quickly, and that kind of steamrolled a lot of response and preparedness because we had a good idea of what the virus was just by doing the sequencing and then follow-up testing as well and analysis. But really trying to kick genomics into high gear as public health means getting out of the big scale and going to local levels, going to individual hospital systems, going to states, going to regional departments. And that doesn't mean just talking to one person at a time, right? There's definitely companies that have amazing genomic technology that want to break into these markets as well. Can we facilitate those conversations so that their technology is better leveraged, right? Can we help support different agencies or entities or public health companies or whatever that don't have their internal genomics core that they can do analyses for? Can we kind of outsource that a little bit and support them while also helping them build that up as well with some corporate strategy? Yes, absolutely. So we see ourselves as multifunctional. We want to be the kind of the multi-tools that
Yeah.
have the familiarity in genomics and public health to fill these gaps. But I think the reflecting on SARS-CoV-2 versus monkeypox versus other diseases, there's definitely been a trend where genomic epidemiology has become more mainstream. The communication has become more effective. Now let's get into that next order of magnitude of implementation by going more local and by connecting with individuals who aren't just genomics gurus, who can benefit from genomics technology in their public health work.
God, I love that. And that's what the epicenter does.
Yep.
Did you have any other questions, Jeffrey?
I don't. I
Okay.
don't. This will be. I think this is going to be exciting to see how
Yeah.
how this venture works out for Dan, and what all we're going to get to learn in the future. As Dan gets the chance to pull all of this together, work with
Mm-hmm.
multiple different stakeholders, and I think I think we're definitely going to be entering a new era of information on
I
how
hope so.
infectious diseases function and how we message for them. And it's exciting for me because one, leveraging technology that has become so much better, so much faster, so much more accurate,
Right.
but number two, leveraging knowledge in order to get that information discussed in the proper way.
Right. I
Yeah,
think it comes
creating these teams of folks who aren't just genomics people, right? Getting
right.
people out of their silos, creating these multifunctional teams, and being the facilitators of that. I think that's a really important goal for us to establish. And
Creating a new language out of two languages.
yeah, or at least connecting the two to be able to interpret, right?
Yeah,
And I
yeah.
want to give a huge shout out. Like, obviously, I'm a key part of this, but I have two amazing colleagues, Alex Sunderman and Mustafa, who are outstanding as scientists. I've worked with them for years in kind of a translational research setting. I really trust them. They have been incredible public health scientists. They are innovative. They are courageous, and they have done lots of really exciting and interesting work. I mean, Alex has a background not just in genomic epi, but as a hospital infection preventionist. Has worked as a public health consultant in the past, and he has this kind of this business strategy mind. There, he's implemented genomics in public health. He's very well connected in the field and very well respected because of his innovation and his willingness to create new opportunities. And Mustafa, he was trained as a physician in Nigeria, in his home country, and then he came over to the United States to train in epidemiology because of the trends in infectious diseases that were happening in West Africa that were affecting his home country.
Sure.
And he's built up this expertise, and he learns so much so quickly. He is the closest person to a guru that I think I've ever met. He is always so wise. He has incredible. Data analytics training that he's built himself. He's taught himself R, like the programming language R. He's always willing to share what he knows and offer some new perspectives. So I think between the three of us, we have some exciting skills to offer, and we play off of one another really well. But I really just want to give credit to the team, because it's not just my effort, right? This is a team effort with some great public health professionals that want to do something daring and transformative in the field. And we're already getting some traction from it, despite it being a very new company.
Of course you are. It's needed, it's valued, and it's something we went through this phase of, and I'm going to simplify it a lot to wrap it up. We decided that we shouldn't test so we didn't know, or we should test so we should know. And that's where the divide in America was, and I find it fascinating that people wanted to put their heads in the sand when there's groups like you getting started to tell us ahead of time. It's not you, it's your neighbor, and you could get it if you don't know. So I believe in this surveillance. I believe in genomic surveillance so that we know ahead of time what's going on. I don't ever want to have another COVID pandemic again that could have been stopped if people had just been paying attention. So that's
Yeah.
where I appreciate you from.
Yeah, there's definitely, like I've said before in the show before, there's going to be another pandemic worse than COVID. We just know that's going to happen, right? So if we can prepare public health infrastructure to be ready to absorb that major impact, part of that equation is implementing 21st century technology for 21st century public health surveillance. And the answer for that is genomics, using genome sequencing, getting that engaged. Not just siloing it to the computational biologist, but really making those
Right.
connections, making policies and streamlining response and preparedness protocols using this new technology, getting people engaged, getting people bought in, right? I think it's going to be a key part of preventing or mitigating the next pandemic when it comes along. And I think in between now and whenever that is, hopefully later than sooner, but there's a lot of ways that it can be implemented in the diseases that we face every day, right? Foodborne infections, healthcare associated infections that happen every
Yeah,
day and kill thousands and thousands of people. There's applications for it right now, and we really want to
right
be those
now.
connectors, those facilitators that bring people together to forge solutions based on this amazing new technology that has just absolutely exploded in the last 10 years.
Agreed. I think you say there's going to be a next COVID, and I think there might have already been one that has been defeated because of people like you, because of the work that you're doing, because of people that actually research and take seriously the science behind viruses. And so we don't even know what COVID pandemics have been knocked down because of the work that you've done. So we got to think about that. COVID was a missed opportunity, but scientists all the time are catching things in the early stages and addressing them.
An
Well,
ounce of
that's
prevention is worth a pound of cure, as
Thank
Benjamin
God.
Franklin
Yes,
said.
America. Yes, America. Let's catch it early instead of freaking out later.
yep.
Thanks for being here, Dan.
Absolutely. Love supporting the show. Thank you guys for giving me some time to chat about my new venture, and I'll definitely keep you all posted and would love to come back and give an update at some point if you'd have me on.
You're always
Of
welcome back.
course.
We have so much to talk about just besides that. But everybody should go to Epicenter Genomics on Twitter, or you can go to Public Access Pod on Twitter and you'll find their tweets, and then you can follow them. They have some interesting stuff. Cool. Thank you.